Welcome to Brainstorm, where we give you a glimpse into the world of science, for this Monday, August 27th, 2012. We begin with a story from the world of neuroscience. A research team headed by Michigan State University has linked a common parasite to suicide and depression, called Toxoplasma gondii. It's a parasite that normally infects cats and was thought to be dormant in some humans, but recent research has suggested that it can actively infect people. This latest study just strengthens support for a link between T. gondii and behavioral changes, particularly depression and increased tendencies towards suicide. Before you go giving away your cat, the parasite's eggs can also be in contaminated water and use other mammals as hosts. In humans, a T. gondii infection seems to cause inflammation in the brain, which itself has been linked to depression and suicide. The inflammation can cause changes in neurochemistry, specifically reduction of the neurotransmitter serotonin. Now, this does not necessarily mean people with T. gondii automatically become depressed, but that an active infection of the parasite may cause inflammation and worsen symptoms of depression. Direct measurement of suicide assessments show that T. gondii positive people scored consistently higher, an approximately 7 times increase in future suicide risk. While this might freak some people out, this is actually good news. Depression and suicide is a serious health issue, and identifying relatively clear biological causes make it easier for treatment development. Certainly not for all cases, but targeting this parasite could be the difference between severe and moderate depression, maybe even saving lives. Our next story is from the world of biology. If you've been keeping up with medical news over the past week or so, you've probably heard about a new male birth control pill. But here at Brainstorm, we like to cover stories you probably haven't heard of. For example, Yale and Harvard developing male birth control for mosquitoes. Now you may be wondering, why do this? But sterilization of insects is a proven strategy for controlling populations. It was first used on the screwworm flies, mainly in the southern United States, as they were costing millions of dollars for the cattle industry. This technique has also been effective in controlling a fly population in Africa known for carrying disease. Luckily, the mosquitoes that transmit malaria have a unique mechanism in their reproduction, which is being targeted in the sterilization research. You see, these mosquitoes mate in airborne swarms, so the males create a plug after mating to essentially keep their sperm in. There's a particular enzyme inside the mosquito's semen that is responsible for coagulation of the mating plug, and blocking this enzyme effectively stops fertilization. The next step is screening for compounds that inhibit the enzyme, and then developing that compound to reduce the mosquito population. Only female mosquitoes bite people and spread malaria, meaning males could be bred in controlled environments and given the inhibitor before being released into the wild. Some of those males would unsuccessfully mate with females, hopefully leading to an overall reduction in the population. Malaria is a serious public health issue, and this method of control has certain advantages, such as being specific to these mosquitoes. These deadly disease carriers are already becoming resistant to common pesticides and changing their behavior to avoid bed nets and other prevention measures. Development will continue, perhaps combining sterilization with other techniques, like genetic engineering. Finally, from the world of evolution. Normally, when we talk about evolution, it's a discovery about human evolution, or the last universal common ancestor. However, scientists at the Fred Hutchinson Cancer Research Center have been studying the evolutionary mechanisms of pox viruses. Probably the most deadly virus from this group was smallpox, and although it was officially eradicated, it has plenty of existing relatives that still affect humans and animals. Pox viruses are interesting because, despite being DNA-based, they're quite virulent and adaptable. DNA is a pretty stable molecule, so pox viruses and other viruses based on it generally have a low mutation rate. But pox viruses frequently cross species barriers and develop drug resistance. To study how they have such adaptable capabilities, the vaccinovirus was introduced into human cell cultures with some modifications. Cells have a particular protein that has evolved to defend against poxvirus infection, called protein kinase R, or PKR for short. In response, poxviruses evolved two genes that interfere with PKR. So the scientists deleted one of the genes. They normally work together, so this modified strain was quite weakened. But it quickly became effective at countering human PKR once again. What they discovered was that the virus was rapidly selected for having multiple copies of the remaining gene. This increased production of that viral protein, somewhat compensating for the missing gene. Not only did this give a quick evolutionary advantage against viruses with less copies, but also provided more material for evolution to act upon. Mutations still happen to the viral DNA, and once a beneficial one appeared, smaller genomes were selected for. Understanding how poxviruses can adapt so quickly should help scientists develop more effective antiviral drugs, perhaps inhibiting gene expansion. Well, I hope you enjoyed this video. If you did, please consider subscribing and be sure to check the links in the video description.